It's my pleasure to introduce John Stockie for the first speaker this morning. And then he's from Fraser University. Yes, I know that.
Not FSU, SFU. So thanks to the organizers for having me. This is an extreme pleasure, especially to celebrate the 60th birthday of our distinguished Ricardo Cortez. And I'll try to tell you a little bit about, before I start speaking, a little bit about why I'm actually here. But first, what I'm talking about today, I just want to mention is the work mainly of an MSc student that I had. It's my first foray into swimmers. And this is work that actually never made it out into the literature. And so I'd be happy for criticism, anywhere from constructive to extreme. You're happy to tell me that this has all been done, but I'd be happy to hear feedback, and maybe I can tell you something different about swimmers that are not swimming in Stokes flows. So this is an outline for my talk. It's based around 4 simulations of various swimmers that are going to look very familiar to people like Fauci, because I've sort of followed some of your earlier work on worm-like swimmers. But I really want to start with a sort of an introduction about why I'm here. So it is an extreme pleasure to be here for Ricardo's 60th birthday celebration. We actually met a long time ago. Long is relative, but for a lot of you, it'll seem like long. I met him at an IMA workshop. We actually spoke at an IMA workshop back in 1999 when I was a postdoc. And Ricardo and I were talking the other day about one of the biggest memories you have of Minneapolis is how cold it is in January. So that was one of the things. But I also remembered Ricardo. And it turns out Lisa was an organizer of that workshop. So
I love it.
You did? Okay. But it's actually possible that we encountered each other. I'm not sure how much we spoke, but at an MSRI workshop back in 1998, celebrating another famous 60th birthday of this guy here. And I just want to mention, I was looking for a picture of Ricardo and Alex Chorin, and this came up with another famous guy in the middle. But it's on this website that has an incredible story about Ricardo. And I mean, I don't want to embarrass Ricardo, but you put it out there. So I learned a lot about you by reading that. So I can't say enough. I mean, you're an amazing person. I mean, that's sort of a little bit of Ricardo's history. So I would encourage you to read that. But we immediately recognized common interests through papers and sort of things we had done. But this paper was an important one for me with Douglas Varela, who's the other guy with white facial hair sitting next to Ricardo. And Ricardo had this inspiration to invite Douglas and myself to Tulane in 2002. So at least we're in the 2000s now. Thankfully, this was in May. So I should have put a sweating emoji there. Even in May, it's hot for a Canadian. But at least I didn't hit the July-August humidity in New Orleans. But Ricardo's idea was, I think this was your idea, but to sort of extend this paper I pointed out on the previous slide on inviscid elastic membranes to viscous flows with a parametrically forced membrane. And this was like a lot of fun to do. Highlights of me for that trip were Douglas drilling us on flow K theory. I learned something really new that was actually a neat tool that I've used in many years since, and deciphering these notes of Charlie Peskin that Ricardo got his hands on. So you remember that, this linearized inverse boundary notes. And plus, we found time for this swamp tour. And Lisa and Ricardo introduced me to soft-shelled crab, which is still one of the best meals I think I've ever had. So just highlights for me of New Orleans. But after years of work and many, many emails and another visit to Tulane, we finally nailed this paper, which unfortunately I am not going to talk about today, but it was a lot of fun and was a source of many problems for me and my students and postdocs over the years. And then I met up with Ricardo at numerous conferences since then. Banff, maybe Toronto. Were we in Toronto as well? I'm trying to think, but definitely Banff. And Ricardo and Lisa generously invited me again in 2016 when I got to show my family beautiful New Orleans too. And we redid the swamp tour on a boat this time and close up of the alligator. I think is that an alligator or crocodile? I never know. Alligator. And we saw some of the sites. So it was lots of good times. I just want to say thanks, Ricardo, for the great times mathematically and, otherwise. And for your willingness to communicate code. I looked through a lot of my old emails, which I still have, and a lot of them are just MATLAB codes. So we have this neat way of communicating in code. And for being such a gracious host. Thank you. So, okay, now I have to give my talk. So I might have to compress this a little bit. So a lot of people study swimming at many different length scales. And there's, I would say, a lot of work has been done on microswimmers in the Stokes regime. Reynolds number is very close to 0. There's been a lot of talks, or there have or will be a lot of talks about this Stokes regime with the method of regularized Stokeslets. A lot of work on CFD simulations of macroswimmers, where there's much more focus on advection-dominated flows. And a lot of recent work on nanoswimmers, and especially things like these nanorobots and things at extreme scales when molecular dynamics dominate. But there's kind of a gap, and I'm not saying there's been nothing done here, but a relative gap in the area of small but still significant Reynolds number. And I'm just going to call this the meso scale swimmers or meso swimmers at this intermediate Reynolds number. Things like longer nematodes or small jellyfish-like creatures and things like that. And so as I said, there's been a lot of work at very low Reynolds number. A lot of these use Stokesian or other approximations. But there is some slender body theory for undulating fish and eels and things like this. And numerous simulations use the full Navier-Stokes equations. They're often restricted to one or a few swimmers. And it turns out there is a recognized gap. I mean, there's this paper, for example, by Wang and Ardekani. It's a bit older now, but despite the wide implications of swimming with inertial effects, these effects of inertia on the motion of small organisms are rarely discussed in the literature. I think this is being somewhat filled now, but at the time I was looking at this with my student, we thought this may be a place to sort of spend some time looking. So in a more recent paper by Daphne Klotza, she made the statement that the collective behavior of mesoscale organisms remains largely unexplored. And so this is what the sort of area that we're looking at. And so our focus was on simulating swimmer dynamics and interactions at this mesoscale. And we called these mesoswimmers. We have some motivations for this, sort of long term. But I would just say this term mesoswimmer, we use this term, Said used this in his thesis, but it's actually appeared since then in the literature. So mesoscale swimmers and mesoswimmers has been starting to appear a little bit more often. So I'm looking at an organism like the nematode, where there is certainly no turbulent flow, but where inertial effects can have a significant impact. I'm, sort of aiming this eventually towards, the study of active suspensions. They primarily, I believe, move by undulatory swimming. And one thing that'll come up in one slide is since the ultimate aim is to look at simulating large numbers of these organisms, we were actually looking at parallel scalable algorithms. And so the nematodes seemed like a good model organism for this. They make up, it turns out, I mean, I think I pulled this from Wikipedia, but they constitute 80% of all animals on the planet. So that is a fact I didn't know. It's not something you think about. They live in the soil, the water, fresh and ocean water, and you typically don't see them because they're so small, but you do have longer nematodes, which is probably more in the range of where this talk focuses. And so this study is really a first step towards reproducing qualitative behavior of these type of nematode swimmers in semi-dilute suspensions, where you get sort of not single swimmers, but some of them are swimming on their own, some in pairs, some collected together, but it's semi-dilute suspension, but certainly not dense. And developing some quantitative measures of the aggregate solution behavior, and trying to identify at least some simple emergent behaviors from when you put some swimmers together in a solution, how they behave. So this is the model problem we're looking at, the standard, doubly periodic 2D domain, swimming sheets. We're not really looking at nematodes. We're looking at swimming nematode-like sheets. Each swimmer, so if we're looking at multiple swimmers, we look at individual curve segments. And the advantage to this type of thing is we can put down an equally spaced grid on the X and Y coordinates that's fixed in space because it's a rectangular grid. And then we, in the immersed boundary sort of manner, we discretize on an equal space, on an equal grid in some arc length parameter along each swimmer. And we're solving the Navier-Stokes equations to govern the fluid dynamics in the immersed boundary framework. There's the singular force term. We've seen this lots, so I'm not going to explain that. And the no slip condition on each of the swimmers is a velocity along the swimmer location, but you can write that in terms of the convolution of the fluid velocity with the delta function. And so the delta functions, of course, well, I don't have to talk about this after Boyce's long discussion of delta and many other people talking about this. But the force exerted by each swimmer, I think, is identical, or at least very similar to what Lisa Fauci and Sarah Olson did in this paper in 2015. We combined a stretching and compression resistant force that where the distance between successive points along each swimmer has some resting length, little ls, and a stretching stiffness, they're called CS here, and then a similar bending resistant term, simple bending force with a bending coefficient CB. And we have to apply some target shape for these undulatory swimmers to say where, what are they, what is their shape? We have to say how they're actually swimming. And this, I think, is taken directly from your paper, Sarah and Lisa. So this is nothing new. I want to say something quick about the algorithm. This was, I tried to get Jeff Wiens to use Boyce's code. I mean, I've talked to you about this before, IBMR, and Jeff was really a computer scientist at heart, and he wanted an optimally scalable parallel algorithm. And so he decided he had to do it himself. So it took a while, but he used this pseudo compressibility method of Guermond and Minev. It reduces the Navier-Stokes to tridiagonal linear systems. He used domain decomposition. I mean, the rectangular geometry made that very nice. I don't want to talk about the details, but basically this is the figures from his thesis, who essentially showed that, in short, that this is pretty close to the best that you could expect in terms of parallel scaling for this. So at least sort of for simple problems in 2 dimensions, we were able to get very, very, very good results. He had some simulations in 3 dimensions as well, although we never used this for simulating swimmers. So I want to show you a couple results from this. So These are the parameters that we're using. Again, I think these were taken from either the Fauci and Peskin paper or Olson. So that's
There's something very special about 0.2 centimeters.
yeah, but we did it. I didn't question, So this was in terms of validation of the method to see if we could reproduce the results that were already out there. And there was this nice asymptotic expansion from Tuck that was in that paper as well that we compared to. And, this will look familiar, Lisa, the swimming sheet going to the left, right? And this figure, this is the data from the Fauci and Peskin paper. And we managed to get a pretty close match to it. So we hoped at least that we were doing the swimming sheet problem properly. So then we moved on to a single swimmer. That's, I believe, a little closer to a nematode. And we varied the beat frequency, and I would just claim, this is very brief claim, but we observed qualitative agreement with experimental observations and other simulations. At higher Reynolds number, the swimmer sheds alternating vortices like you would expect, and the amplitude of the oscillations of the tail increased with the Reynolds number. These are all things that you'd expect to see. And here are a couple of, these are increasing time and increasing Reynolds number, where you can see the behavior of these swimmers. And I'm just, I spent so much time talking about Ricardo. I'm just not going to discuss these results too much. We just had pretty good qualitative agreement, okay, with other simulations. But the thing I wanted to focus in on is this picture that is a plot of the swimming speed versus the Reynolds number. And the swimming speed increased with Reynolds number, except for this small blip around Reynolds number 2.5. So there's this little, somewhere around here. So a possible explanation for this, we don't have anything definite to say here, but for smaller Reynolds number, the viscous forces dominate the production of thrust. Whereas for larger Reynolds number, the forward motion is driven by the momentum in the wake of the swimmer. This is a paper that discussed this idea. And Brackenbury also claimed that the vortices intensify and shift into the wake as Reynolds number increases, and we see this in our simulations. And so this could be a possible in this sort of transition regime where that could explain something like this. And if we modified the parameters and looked at this kind of the other parameters other than Reynolds number, this was like reproducible. So it didn't seem to be a numerical issue. Okay, so we don't have a very clear explanation for that. So if this explanation is wrong or someone has a better idea, I'd be happy to hear about it. So this is, like I said, we did not put this through peer review yet, right? This project actually got sort of delayed, and then eventually stopped due to COVID. This was one of those students that actually finished around COVID time, and this is a project that's been hanging since then. So going back to the Olson and Fauci results, there's this problem they looked at with out-of-phase swimmers that are synchronized. And they observed that Stokesian swimmers synchronized when having this phase shift of pi over 2. And we see similar behavior at smaller Reynolds number, close to 5 or less. But at higher Reynolds number, the synchronization can fail. And we believe this is due to larger hydrodynamic forces that actually repel the swimmers. So you get this behavior where they look like they're starting to synchronize and aggregate, but then they whip apart. So nearby swimmers, it seemed to track each other as long as the Reynolds number is small. And this picture is a plot of the head and the tail difference as a function of time for these two swimmers, just to show you that happening. So that's something that happened, not all the time, but sometimes. And then we cranked this up to simulations of larger numbers of swimmers. I think we had up to 100 or 200 swimmers. And we initialized the domain with random location and orientation. And then in order to study the behavior of the suspension, since we're aiming towards looking at sort of semi-dilute suspensions of large numbers of swimmers, we were looking for some way to measure the, are swimmers close together or not in an average suspension sense. And so I did, as a PhD student, I did some work on pulp fiber dynamics, actually. And so from the pulp and paper literature, there's this thing called, I don't know if you've heard of this before, but there's a pair distribution function for the distance between pairs of particles in a semi-dilute suspension. And this is it. And we applied this to studies of these swimmers. This initial distribution gave a pair distribution function that looked something like this, where you see some peaks where there's certain distances that particles tend to, where the minimum distance between them is there's certain peaks in that distribution function. And so these are plots at increasing time. So these are snapshots of the suspensions as these swimmers are aggregating and then separating and moving apart. And it turns out the aggregation and local alignment both peak around R is about 2H. So in the immersed boundary method, that's actually the delta separation distance. So we can't really get them much closer than that. So that's kind of a numerical artifact. But there is this peak that says they do get very close together, but then there's, some others that swim close to each other, but not within that almost touching distance. So this just gives you an idea of how this picture of the suspension turns into this pair distribution function. But then so that was for Reynolds number 5. If we increase both the number of particles and the Reynolds number, we actually had a different behavior in this aggregation function. So the aggregation actually happened more rapidly, probably because they're packed closer together, but it's also transient and less intense. So, as you can see, this leaves a lot of questions open, but that's where my talk ends. Because this is where this work ended. There's a lot of things to answer. Why is this behavior seen in denser suspensions? Can you say something more about the intensity of the aggregation and things like that? So this is sort of where things are at the moment. So just in terms of summary, we validated our two-dimensional immersed boundary model for these undulatory mesoswimmers with other both asymptotic and numerical results. We observed significant inertial effects that seem to mimic actual active suspensions. This synchronization and phase locking, enhanced swimming speeds, different attraction and repulsion behavior, this alignment and aggregation in the pulp and paper literature, that's called flocculation. I think, do you use that in swimmers as well, flocculation? Not really. It's usually for passive particles. And this is even at relatively small Reynolds numbers. So there's actually quite a range of these, intermediate Reynolds numbers where this seems to be an issue. And this pair distribution function I'm claiming could actually be useful in the study of active swimmers as well. So, I would say, where is this going? Well, right now, until I get a student who can take this over, maybe it'll just sit for a little longer. But the observations are primarily qualitative, and we'd like more direct comparisons if possible. Wall interactions with these nematodes certainly have some interesting dynamics, and you can see that in this paper by Yuan. The limitations in 2D were actually simulating flapping foils or sheets instead of worms. And there's things like turning dynamics where these nematodes tend to cross themselves in order to turn that we couldn't possibly simulate in 2D. So there's certainly a value of doing 3D simulations here as well. So that's basically it. Thank you.
Are there any questions?
Yeah.
I want to ask, first of all, a really interesting talk. And I want to know if you want to have two sheet swimmers, do you have repulsion forces between them in the model?
No, we didn't. They tended to, well, if they're far enough away, they just kind of swim independently.
But when they close,
As soon as they got within a certain distance, they tended to aggregate in phase lock. That was.
They attract.
Do they attract? They tended to move a little closer to each other. Yeah. They never touched, but that's an inverse boundary thing, right? The deltas don't allow them to touch. So they never quite touch. But you have to think the delta function introduces some kind of effective thickness. So it's essentially like touching, but we didn't introduce any specific attraction forces or anything.
so time step is consistent through, like the same through the round. Change your pattern when they
Time step is constant. Jeff's code was actually a variable time step code. So we had some adaptive time stepping in it. But when you have these swimmers, like the time step doesn't vary a lot for these particular simulations.
Okay, got it. Thank you so much.
Yeah.
Okay, anyone else?
Were they identical? Did you have some fluctuations in the frequency and?
The pictures I showed you were identical, but we also did simulations where there were small random. Oh, sorry. Let me just look at my slide and make sure. They had random location and orientation, but the frequencies were the same. Yeah, most simulations. But then Saeed did some playing around with the like random amplitude and frequency of oscillation to say we had swimmers with different dynamics. It didn't make a huge difference, but this is, again, COVID hit and we had to leave that as is. So this would be interesting because I guess nematodes, I mean, they're all the same species, but they have different gates, some variation. Yeah.
It's not really a technical question, but do they, like when they aggregate, are they like secreting any like viscoelastic like substance or like mucus?
Sorry, I'm a mathematician, not a biologist. I know nematodes from pictures. That's all I know. They could be, and I mean, like, how do they aggregate, right? I mean, some of this could, like, I don't know how smart nematodes are. Do they sense each other? And actually, they do.
I mean, they have sensory.
Yeah. So I built in no directional ability or anything, and I mean, we all secrete, right? So I'm sure they secrete stuff, but I have no idea. Maybe I should stop there before I ask anything else off color. Yeah.
No? Okay. Any other question? Okay, let's thank our speaker, John.